Thank you very much for inviting me. I believe my scientific collaborator, Don Siegel, has also joined the meeting because he is considerably interested in this project, this technology, which he originated. This project grew from Dr. Siegel's deep scientific and personal friendship with this scientist, a scientist who died too young in 2014 from medullary thyroid cancer, a scientist who may well have won a Nobel Prize for his groundbreaking technology for identifying targets for cellular and other therapies, Dr. Carlos Barbas. This is just a picture of medullary thyroid cancer, which you've probably seen multiple times during this symposium. It arises from parafollicular or C cells in the thyroid gland. Many medullary thyroid cancers are as the result of RET mutations. But one thing I want to make clear as I start this presentation is that the therapy that I'm going to tell you about is a therapy that is suitable for people with and people without RET mutations. But in this particular case, Dr. Barbas did have a RET mutation, and this is just a picture showing the RET gene, which crosses the cell membrane and signals inside the cancer cell. And in this particular case, this RET mutation led to the fact that this RET gene was always on. So it's like a stuck accelerator. The idea was, on Dr. Siegel's part, to use the scientific method designed by Dr. Barbas to try to identify a target that could be used to generate a cell therapy for Dr. Barbas himself. And so the idea was to use Dr. Barbas's scientific method to identify a target, and that target might be specific to Carlos's tumor, or specific to medullary thyroid cancer, or just a target that would be suitable for attacking C cells. The idea then was to make an antibody to that target and use that antibody to try to target medullary thyroid cancer cells. The goal of identifying this target was to generate a chimeric antigen receptor T cell, or a CAR cell. Chimeras in Greek mythology were creatures that were a mix of other creatures, including the head of a lion, the body of a goat, and the tail of a snake. Now, in the lower part here is a normal T cell. These are white blood cells in your body. These T cells all have receptors in them. That's the blue in the cell membrane and the purple stuff inside. And the idea was to take these T cells and re-engineer them, modify them with a different kind of receptor that would recognize a particular target. And so the idea here was to use Carlos's technology to identify a target, make an antibody to it, and that's shown here to the left, and take parts of that antibody and put them into a human T cell, in this case, a T cell from the patient, so that this T cell would now recognize this target and kill it. And I will come back to this a couple of times because it is a complicated concept. So Dr. Siegel identified a bunch of targets using the very technique that his friend Carlos had developed. This slide has print that's very small, and that's intentional because I don't want you to read all of it. But these are some of the more promising targets that were in the mix. And one of them, GFR alpha 4, seemed to be, for a variety of reasons, some of which I will show you in a moment, to be particularly suitable as a target for this particular methodology. So what is GF-alpha-4 and why is that important? So this is a generic slide that is just talking about choosing targets for CAR T cells. And GFR-alpha-4 turns out to be a really unique and special target. What you're looking for in theory is a target that is only found on cancer cells, not found on other cells in the body, not found on other organs in the body, and also dispensable. So in theory, you could attack the target, eliminate it, and anything attached to it, and nothing terrible would happen to the person because you would only kill the cancer bearing the target and not damage other cells in the body. So it turns out that GFR-alpha-4 is very specific to C cells, and these are the parafollicular cells that go rogue in medullary thyroid cancer. And on the left here is a blot just showing that GFR-alpha-4 only shows up in the thyroid gland and is not found in any of these other tissues going across the top of this blot. And this is just a slide showing you that the brown staining against GFR-alpha-4 only occurs in medullary thyroid cancer cells and does not occur in other types of tumors. This is a very critical panel just emphasizing that GFR-alpha-4 is not found in other parts of the human body. It is only found in the thyroid or in medullary thyroid cancer cells. So it has absolute specificity for medullary thyroid cancer cells. So Dr. Siegel, now equipped with this knowledge about GFR-alpha-4, wondered if he could make a CAR T-cell therapy against GFR-alpha-4 for his friend Carlos. So just to recapitulate, you identify a target using these methods for creating new antibodies that Carlos had taught Dr. Siegel years earlier. Could you now use that information to make a CAR T cell? You've seen this picture before, just emphasizing the structure of these CAR T cells, that you're taking T cells from the body. They normally have these so-called T cell receptors in them, and you're replacing those or adding T cell receptors that are now directed against a particular target. So how are these CAR T cells made? They are made, and they are made by collecting blood from a patient, purifying T cells from that blood sample, go to the lab and put this new receptor into these CAR T cells, generating these CAR T cells, which are then expanded in a dish over a period of a couple of days to a couple of weeks, subjecting them to quality control to make sure that they're not infected or anything like that. And then they are infused back into the patient, where in theory, they will go after the tumors and kill them. And so this is a cartoon that is animated that is showing this man-made T cell receptor containing this antigen binding domain that just recognizes specifically GFR-alpha-4. It is a modified T cell receptor, so it has this signaling apparatus inside the cell, which is very important for the activity of T cells, which are designed to kill cells infected by viruses or cancer cells. This is a cartoon showing that this T cell receptor, let me go back a moment, this is the normal T cell at the top. We have now introduced these man-made T cell receptors that are directed against GFR-alpha-4. And the whole purpose of this modified engineered CAR T cell is to go after the cancer cells and kill them. This therapy is designed to be infused once, and the infused cells ideally are capable of dividing in the body and persisting in the body, perhaps sometimes for years. And this is just another cartoon for those of you who are visually oriented, just showing the CAR T cell at the top, the ugly looking cancer cell at the bottom. The CAR T cells are going after this particular cancer cell, which in this case, let's pretend that it's an MTC cell bearing this GFR-alpha-4 target. The CAR T cells have been designed to recognize that, and they kill it. So the way all of this technology started was a fairly long time ago, about 14 years. showing that this could be done in patients with chronic lymphocytic leukemia. New England Journal of Medicine article, and additional scientific articles showing that the promise of CAR T cells was in part that they could stick around and be in the human body for a long time, continually killing any cancer cells that might arise. This is actually the very first patient who was ever infused. This patient is still alive. This patient had a disease called chronic lymphocytic leukemia. He was infused with CAR T cells, and this just shows his bone marrow, which was loaded up with these blue cells, which are bad. And when all was said and done and his repeat bone marrow was analyzed, it didn't have any evidence of these abnormal cells. The very first patient with ALL was a patient named Emily Whitehead. And by the way, these patients have given permission for their identities to be revealed. She was 5 years old when she was treated. She recently matriculated at Penn in the class of 27. This gentleman here is Dr. Carl June in the middle of that picture. And he is the scientist whose brain this particular concept of CAR T cells came out of. So getting back to Carlos and GFR alpha 4 directed CAR T cells, there were a number of experiments that were done in the laboratory demonstrating in various mouse models that these CAR T cells would work. And this is just an experiment showing the ability of these CAR T cells in the lab to kill medullary thyroid cancer tumors. This was also true in a mouse model where the animals were injected with thyroid cancer cells. The tumors were allowed to grow, and control normal T cells didn't do anything. The CAR T cells killed the tumors. That's why these graphs are headed south. And this is just showing that the tumors would shrink. And these are pictures of mice with the medullary thyroid cancer cell tumors in their flanks, showing that after they were infused with these modified T cells, the flank tumors basically were killed and disappeared. To date, CAR T cells have only been successful in liquid tumors. These are some of the CAR T cells that are FDA approved for the treatment of leukemia, lymphoma, and myeloma. These are the CAR T cells that are being directed against solid tumors. They're all experimental. None of them are proven and none of them work. Solid tumors like medullary thyroid cancer are a huge problem because of target selection. Remember, GFR alpha 4 is special, but many of the targets are not special. They're not completely specific to cancer. They're found on other normal cells. And if that happens, the CAR T cells will attack those normal organs and kill them with adverse consequences for the human host. There are a number of other technical challenges for CAR T cells in solid tumors. Liquid tumors are accessible. They're in the bloodstream. It's easy to find them. It's easy to kill them. Solid tumors are hiding, embedded in organs like lymph nodes, bone, brain, et cetera. And the CAR T cells have to execute an obstacle course. This is showing a CAR T cell in the bloodstream. It's green. It has to get out of the blood vessel. It has to negotiate a path into the middle of the tumor in order to kill it. And that is not easy to do. And the tumor also has a number of defenses. Let's call them minefields, moats, trenches, a variety of ways of making the CAR T cells exhausted, preventing them from getting in the first place, or the cancer cell simply eliminates the target that the CAR T cell is directed against, or it has an environment inside the tumor with a variety of cells that are hostile to the T cells that we are trying to get into those tumors to kill it. So this is our clinical trial. And just a brief summary of the trial is that we are looking at the safety of CAR T cells. We want to demonstrate that manufacturing them is feasible and describe any preliminary anti-cancer activity. We want to know, do the CAR T cells end up in the bloodstream and for how long? Do they actually get into tumors with biopsies to show that? And we're measuring a variety of other immune factors in the bloodstream that should be activated if the CAR T cells work. We're looking for adults with medullary thyroid cancer that has been treated with prior therapies or the patient has declined to receive such therapies. Basic organs like kidney, liver, and so forth have to work. The patients have to have what we call a reasonable performance status, functional, and they have to have something that we can measure, a tumor that we can see on a PET scan, on a CT scan, calcitonin, and so forth. And there are a variety of very standard exclusions for this trial, including uncontrolled heart disease. Steroids are bad for T cells, so we don't want people who are on high doses of T cells. And we don't want people who have active autoimmune disease requiring immunosuppressive therapy, because after all, our CAR T cells are an immune therapy, and we don't want to activate autoimmune diseases. We are giving a single dose. There are 3 cohorts, 3 different doses. We are currently in cohort 2, and there will be a total of 3 cohorts. The doses in these cohorts are by analogy to the CAR T cells that have been developed for other diseases like leukemia. We do give chemotherapy for 3 days prior to the CAR T cells. What we are doing here is eliminating normal lymphocytes, creating some space, some room in which the infused CAR T cells can thrive and proliferate. And as noted before, this is a single dose of CAR T cells via intravenous infusions. There are 5 phases, including screening, the withdrawal of T cells from the blood, and the manufacturing of the CAR T cells, which can take a couple of days to a couple of weeks. We give the lymphodepleting chemotherapy. We then infuse the CAR T cells, and we follow the patient, and we will follow the patient for a very long time post-infusion. We also have permission from the FDA now, and we have done this to retreat subjects who have demonstrated clinical benefit after their initial infusion. We can infuse them again, because when we do the manufacture, we often end up with extra cells that can be constituted into a second dose. We were very worried about side effects. We were worried that we would target and damage a normal organ in addition to medullary thyroid cancer. I already showed you this slide, and I showed you that the GFR alpha 4 target is very special. It doesn't seem to be expressed on other cells in the body. There are a variety of other side effects that we are worried about, including class effects like cytokine release syndrome, neurological effects such as ICANS. And so we are concerned about the potential for these side effects. It is one of the most important aspects of this program. Cytokine release syndrome is a syndrome in which the immune system is instantaneously activated, resulting in high fevers, low blood pressure, and a variety of other symptoms. And this has been seen with other CAR T cells. It can be managed if you're skilled at it with Tylenol, steroids, antihistamines, and so forth. And there are a variety of anti-cytokine therapies such as tocilizumab, cetuximab that are used if necessary. And the University of Pennsylvania has been instrumental in demonstrating how to do this, because we were among the first institutions to ever give CAR T cells to patients with leukemia. The neurologic toxicity is mysterious, but it does occur, and we're worried about that for any CAR T cells. Most of the instances are limited. Brain swelling can occur. It is reversible if it is properly managed. And there are some theoretical side effects that include the use of a virus to construct these CAR T cells. And so we are changing the DNA, the genes of these cells, and you're always worried that you may generate some replication competent virus that may do bad things in the body. We're also concerned that the T cells that we're infusing into the patient may proliferate in an uncontrolled fashion. And we're also concerned about the possibility of cancer caused by the very T cells themselves that we've engineered. That has not yet occurred, but it is an important theoretical consideration. So here's what we have learned so far, and then I'll open it up to questions. We have not seen any off-target side effects. And so this confirms what we thought and what we had hoped about the unique target GFR alpha 4. We have not seen any unexpected side effects. We have seen cytokine release syndrome, which has been manageable, and we have not seen the neurological toxicity. And we have seen some evidence of anti-cancer effects. We have seen tumor shrinkage on scans. We have seen drops in calcitonin levels in the blood. And we have evidence in one tumor biopsy so far that infused CAR T cells are actually executing that obstacle course successfully and making it into tumors. So enrollment in this trial is currently ongoing at dose level 2 and dose level 3. The next patient is due to be infused on Monday, and the patient after that is due to be infused approximately a month later. So at this point, I appreciate your attention. I will open it up to questions. And
Todd Hansen from Houston. We spoke on the phone a couple of weeks ago. And I was anxious to know if you saw any of the results from patient number 4 at the first dose escalation, if you've seen his results or scans or have any input on what has happened to patient number 4.
So this is an ongoing clinical trial. And it is best that data for that kind of a question be answered in the course of a scientific symposium. That's just generally the rules of the game. And so I have been intentionally vague. I'm trying not to be evasive. I'm just trying to be respectful of some of the scientific boundaries. So when you have all of the data, that is when you reveal it to the world. So what I told you in the last slide is, in fact, totally accurate and very encouraging. The absence of side effects is stunning. The existence of cytokine release syndrome is actually encouraging because it shows that something's happening. And it is truly remarkable that with this brand new technology, in the first couple of patients treated, you're seeing any anti-tumor effects at all.
How might the past surgeries affect the decision to administer CAR T therapy and its efficacy?
They really shouldn't. That's part of standard therapy. And so people have gone through surgery, some certainly to remove the thyroid gland and sometimes to remove tumors. And people are coming into a trial like this with worsening of disease, with new tumors that perhaps can't be resected, removed surgically. It really should not have any effect. The most important features of being eligible for the trial is that the patient doesn't have any good therapies remaining and still is in pretty good shape, and of course is highly motivated to undergo, can only be described as a highly experimental therapy. It's important for me to state at this particular time that the patients participating in this trial, they're my heroes. So these are people truly stepping into the unknown with a completely novel technology. We have no idea what's going to happen. They have no idea what's going to happen. That's who the trial is for. So people who no longer have useful therapies, including surgery.
Thank you. How soon after the infusion do you see tumor shrinkage? Do you have any idea of the total percentage of T cells in a person you are transforming?
Again, some of that is very precise data that's still being collected and will be collected. The first look at anti-tumor effects is very standard and is typically at around 2 months. Remember that this is an ongoing trial with a very limited number of people treated. So when I reveal to you that we're seeing anti-tumor effects, we're seeing them at 2 months. A big unanswered question is how long do those anti-tumor effects last? Do they deepen? Do they go away? Do they stay around? So for a trial that is ongoing, we of course don't have the luxury of the follow-up that I showed you with the CLL patient or with Emily Whitehead, where they were treated more than a decade ago. And so we have the luxury of knowing what actually happened to them over many, many years. This is very much a work in progress.
Thank you. If it's in the bone, is it more difficult? Is it a more difficult obstacle?
I think just speaking as an oncologist, tumors that are in bone, tumors that are in liver, and tumors that are in the brain are harder to get to. It's much easier to access tumors in the lung because the lung has a lot of blood vessels. What we really don't know, because it's a very limited number of patients, is whether some sites are better than others. And so the only way that you're going to accumulate those data is by carefully describing in patients, and the patients are all different. Some of them have tumors in nodes, some of them have tumor in node and bones. And you will discover in the course of a clinical trial which sites are more or which sites are less susceptible. But it is really impossible to say right now. I'm just revealing my bias that I think bone tumors and liver tumors are more difficult for CAR T cells to get into.
Okay, thank you very much. Have any of the patients had an objective response? Also, how can you tell if the tumor shrinkage is from chemo or CAR T?
So I will I will answer the second part of the question first. The chemotherapy that's being given, the so-called lymphodepleting chemotherapy, is actually chemotherapy for lymphoma. It is not a chemotherapy that should have any effect on medullary thyroid cancer, which is a notoriously chemotherapy refractory or chemotherapy resistant disease. So if there is tumor shrinkage, if there is a drop in calcitonin, it is very difficult to conclude anything other than that that was due to the CAR T cells. As far as objective responses are concerned, that is classic at this point, unaudited data. And so the way this works is that those scans, and this is actually happening as we speak, are being analyzed by an independent committee, a committee that group of radiologists that has nothing to do with me because I'm biased. So this is an unbiased group that will provide us with a formal report on whether these responses meet the objective criteria for response, which is referred to as RECIST, R-E-C-I-S-T. And then these responses need to be confirmed, which means patients get scanned every 2 or 3 months, really for years, in order to see if those responses endure. So at this point, work in progress, stay tuned.
Is there potentially a study in the works for oncologic cancer of the thyroid and CAR T?
Oncocytic. I think this is, I appreciate the question because it has certainly occurred to Dr. Siegel, is whether Dr. Barbas's technology could be applied to papillary thyroid cancer or follicular thyroid cancer, or even some of the less common versions of thyroid cancer, such as oncocytic, to see whether you could identify targets unique to those cancers that in turn could be turned into CAR T cell therapies for the other kinds of thyroid cancer. What I would say is that MTC is kind of a forerunner because it is leveraging an absolutely unique target. And if we can show that you can use CAR T cells to effectively treat medullary thyroid cancer, that effectively opens the door to CAR T cells for other even less common forms of thyroid cancer, or frankly, the more common forms of thyroid cancer, such as papillary. So I think Dr. Barbas's technology lends itself to the discovery of novel targets. And the hope is that the CAR T cell therapy for MTC will provide a critical proof of concept for the targeting of, well, advanced solid tumors generally and MTC in particular.
Thank you. Does the body learn how to make new T cells that mimic the CAR T cells?
Probably not. So these are fake cells. You weren't born with them. And so one of the most important research questions, and I alluded to that when I presented the information on the CLL patients, is that the CAR T cells for leukemia do seem to be persisting. They persist at very, very low levels, but those low levels are enough for those cells to stick around and perform tumor surveillance, preventing the reemergence of cancer. So that's one of the research questions in this program, is that you will, of course, get this initial burst of T cells in the body right after you infuse them. Not all of those cells will survive, and the hope is that some of them will last and will stick around, perhaps even for the lifetime of the patient, so that they can perform tumor surveillance. But the way the immune system is constructed, we don't have a good way to teach the native immune system how to make these CAR T cells in the body itself. These CAR T cells are made in a lab.
First, Dr. Cohen, thank you for your presentation today and for the work of you and your team. It is greatly appreciated by everybody in this room and everyone we represent. My question is, with the success you're seeing with the limited number of cohorts in the trial now, if that success were to continue throughout all of these cohorts, what is a hopeful time frame and what is a realistic time frame that you would have when a trial could potentially be opened to a larger patient population?
I think for better or for worse, it probably will take years. The issue is going to be funding. So this gets into the whole rare disease funding issue. And with CAR T cells, the funding is a particular problem because the CAR T cells are very expensive to manufacture. Right now, the trial is being paid for by a philanthropist. So somebody has donated money in order to allow this to occur. Rest assured that the whole funding issue and the transition of this trial into a larger trial is really quite are critical issues that we need to think about. I don't have answers for those. And this gets into the whole rare tumor issue. Who's going to pay for these? Is a pharmaceutical company going to be sufficiently interested in this product to fund the trial? Those conversations have occurred. Those conversations are ongoing, but realistically, those conversations and those project timelines are measured in years.
Thank you. Is there any way to monitor or see the progress of the future patients, like the one starting Monday and then the next one the month after that and so forth? Is there any way to monitor the trial, or is that all private?
We are monitoring the trial on a regular basis, but it is not a convention in clinical trials generally, or medical oncology trials in particular, to provide regular real-time updates on a patient-by-patient basis. I've never seen that done. And I think part of the problem is that there's lots of data from the patients, including how are they doing, what do their scans look like, what's their calcitonin levels, what do their T cell levels look like, and so forth. And that data sort of comes in a piecemeal fashion. And what really matters is the total package, and that takes a while to put together. And also, if you really have any hope of going to a federal agency looking for registration of the trial, it's best that you have a complete package available, because that's what the regulators are going to be looking for. And frankly, that's what the NIH grant makers are going to be looking for as well. So they do frown on this piecemeal reportage that might occur.
Thank you, Dr. Cohen. Related to, you mentioned that this treatment is very expensive, in that it's expensive to produce the CAR T cells. Is there any way to compare that to, I mean, cancer treatment in general is very expensive, right?
Yes.
So is there any way to look at it from is this comparable to what current cancer treatments cost? Is it way beyond that? And over time, is this something that could come down significantly in price with, as time moves forward? And lastly, is there anything that we can do to help?
All exceedingly important questions. So in general, CAR T cells are more expensive than other therapies. So the current therapies like pembrolizumab, for example, for lung cancer, are costing on the order of $150,000 to $200,000 a year. And society, both in Europe and the United States, has decided that that's okay, given their efficacy. CAR T cells probably are 2 to 3 times more expensive in general than those therapies. However, if this all works out and it turns out that you have a one and done, where you can manufacture the CAR T cells, you can give them to a patient and you never have to treat them again, then society has decided, with rare diseases like spinal muscular atrophy, that $500,000 is okay. Or even $1 million is okay if you're curing blindness. So I think it's really going to depend upon efficacy. I think if the CAR T cells transiently improve things, but not for more than 3, 6, or 9 months, then the price points that I've just told you about are going to be untenable. On the other hand, if the therapy really has durable efficacy and tumors shrink and patients are doing well, and that doing well is measured in years, then I think society has already decided that it has a pretty high tolerance for high prices under those circumstances. And I think the prices of cancer drugs are indeed very high. I don't know how much higher they can go. And I do think once you scale it, that some of the prices should start to come down a little bit, or at least plateau. So I hope that that answers the question. There's just no getting away from the fact that the technology is complicated. The viral vectors are expensive and difficult to generate.
Dr. Cohen, we really appreciate your time. We were very much looking forward to your session. We do not have any more questions at this time, and we look forward to hearing from you for updates in the future.
Yes, so I
Thank you so
appreciate
much.
being invited, and you can invite me back, and I will update you to the extent that I can. I'm thrilled and delighted that we are now regularly treating and screening patients for this trial, and ThyCa is partly I thank ThyCa for broadcasting our trial, for the membership telling each other about it, and for sending patients our way.
Thank you very much. We will invite you back. Thank you very much.
Thank you.